Good afternoon, everybody. It's with great privilege that I introduce to our moderator this afternoon and co-chair of the workshop, Elizabeth Parks. Elizabeth is a professor in the Department of Nutrition and Exercise Physiology in the School of Medicine at the University of Missouri in Columbia. She also serves as an associate director of the Clinical Research Center in the Medical School Institute for Clinical and Translational Science. The major research contributions of Elizabeth's lab emanate from her developments of novel techniques to quantitate the delivery and disposal of dietary macronutrients in animal models and in humans. She was the first to discover how post-meal processing of dietary sugars being made into fat cause non-alcoholic fatty liver disease. Over the past 20 years, her lab has used stable isotopes, mass spectrometry, and mathematical model to establish a non-steady state model of how fat metabolism changes from the fasting state to after meals. Elizabeth is the recipient of the Robert Levy Award from the Kinetics and Metabolism Society associated with the Arteriosclerosis, Thrombosis, and Vascular Biology Council at the American Heart Association. She is a fellow of the American Heart Association and the Obesity Society, where she is currently on the leadership path to serve as the Society's president in 2022. Dr. Parks' current research is focusing on how an individual response to a weight loss diet results in the liver's recovery from fatty liver disease. Elizabeth, it's all yours.
Thank you, Jose, and thank you for that introduction. Welcome back to the NIH's workshop. Historically, nutrition research has discovered essential nutrients and designed investigations that have focused on a single nutrient at a time. This has led to significant advancements in our understanding of the connection between diet and health. During the past decade, healthful dietary patterns have been identified, and there's a major effort now to understand individual responses to dietary change. The goal of this afternoon section of the meeting is to focus on methodologic approaches designed to understand how people respond to dietary intake differently. This section of the workshop is divided into two parts. We will now start the first part, where we're going to focus on identifying and understanding potential contributors to inter-individual variability in dietary responses. You will hear talks from three speakers. Marian Neuhouser from the Fred Hutchinson Cancer Research Center is going to discuss the importance of examining nutritional status as a source of inter-individual variability. Then Helen Roche from the University of College Dublin School of Public Health will present on immune status, system status, and inflammatory responses to diet. And Frank Scheer from Brigham and Williams Hospital will speak on the effects of sleep and chronobiology. Then Josiemer Mattei from the Harvard T.H. Chan School of Public Health is going to teach us about nutrition-related health disparities, socioeconomic influences, and social determinants of health. And Cheryl Anderson from the University of California, San Diego will speak on the psychosocial and cultural factors influencing dietary patterns. Following Dr. Mattei's talk, we'll have a discussion with these speakers, and then we'll take a short break. After that, we're going to come back for part two, and I'm just going to briefly mention that because we'll start right away after the break. We're going to focus on methodology to assess nutritional physiology and responses to eating. Laszlo Barabasi from the Northwestern University Horry College of Computer Sciences will speak on environmental exposures and response to diet. I'm going to give a talk that will cover various methods to measure the response to diet after mixed meal challenges. And Valerie Duffy from the University of Connecticut will speak on sensory nutrition. When she's done then, our expert panel will discuss research gaps, and we'll end this section by hearing from Saroja Vorogante from the University of Carolina Gillen School of Global Public Health, Krista Verde from the University of Illinois at Chicago, Erin Hanlon from the University of Chicago, and Susan Malone from the New York University Rory Myers College of Nursing. So we're ready to start our talks for this afternoon. Thanks very much for being here.
Thank you to the NIDDK and NHLBI for sponsoring this important workshop. And thank you to Dr. Lynch and Dr. Pratt for the invitation to speak today. My charge is to talk about the importance of examining nutritional status as a source of inter-individual variability. Here are my disclosures. Randomized controlled trials provide a rigorous approach to testing the effect of diet on health. Outcomes may include disease endpoints, which require long-term studies, or surrogate endpoints, such as blood or urine biomarkers, body composition changes, or mammographic density. Controlled feeding studies are particularly useful for testing the effects of diet on biological mechanisms due to the tight control over provided foods and beverages. In the best of all possible worlds, this is what all of our dietary intervention study results would look like. A clear separation of the intervention and control groups, allowing us to draw clear and definitive inferences about aspects of diet we are testing in relation to the outcome of interest, such as a disease endpoint. And in fact, there have been a few such trials. The carotene and retinol efficacy trial, or CAROT, provided strong evidence that high-dose beta-carotene plus retinol supplements increased lung cancer in smokers, as you can see from the figure here on the slide. There was a very clear separation between the active treatment and the placebo. I should mention that the direction of effect was not the intended one, as it was hypothesized that the supplement would reduce lung cancer, but the results nonetheless provide a clear, definitive result. Asking further questions of trials can often reveal inter-individual variability in response that may not be seen with the initial trial results. So the next several slides will provide examples of this phenomenon. The first example comes from a randomized crossover feeding trial where we tested low versus high glycemic load diets on several intermediate biomarkers related to cancer risk. This was a crossover trial where each person served as their own control, and this crossover design reduces some noise and also has the benefit of effectively increasing sample size. Some of the primary study findings were that the low glycemic load diet reduced C-reactive protein and increased adiponectin, and the strongest results were in participants with a high body fat mass. It's important to recognize that even in a rigorously conducted controlled feeding trial, study participants are not a one-size-fits-all. They bring heterogeneity in usual diet, lifestyle habits such as physical activity, alcohol intake, the microbiome, and body composition. More recently, we have been doing further examination of the data from this controlled feeding study. And while we did have significant differences between the study arms for the biomarkers I just mentioned, our recent looks at the data also revealed heterogeneity in response. As you can see here, most participants had an increase in adiponectin on the low glycemic load diet, but some had a decrease. While we do not yet have the answers to why this may have happened, it supports the idea of a heterogeneous response from the exact same controlled diet. The next example is from the Women's Health Initiative Dietary Modification Trial. The study design is shown here on the slide. WHI was a behavioral intervention in over 48,000 postmenopausal women across the U.S., where the intervention goals included lowering fat to 20% of total energy, increasing fruits and vegetables, and increasing grains. One of WHI's primary outcomes was incident invasive breast cancer. And while the results on the left appear to closely mirror ideal trial results with good separation of the intervention and comparison groups towards the end of the trial, the 95% confidence interval for the breast cancer hazard ratio included one. So the results for invasive breast cancer were viewed as suggestive only. Nonetheless, there are several subgroup analyses that may be informative. The first one is shown here and showing that participants who started the trial with the highest percent energy from fat at greater than 36.8% and on average decreased their fat intake by over 12% had a breast cancer odds ratio that was 0.78 or a 22% lower risk, while those who began the study with a self-reported lower fat diet had no such decrease in risk. This says to us that the baseline diet influenced the interindividual variability in response. A more recent analysis was conducted on the cardiovascular outcomes from WHI. And as a side note, cardiovascular endpoints were secondary endpoints to the trial. So here on the left, you can see that overall, there was no effect of the low-fat dietary pattern on incident cardiovascular outcomes, including myocardial infarction, Ph.D. death, stroke, and so forth. However, when we split the group into women who did not have high blood pressure versus those who did have high blood pressure, there was a clear and strong difference in the result. I will also mention that these subgroup analyses were all Bonferroni adjusted, and these contrasts remain striking and strong. So the point is to demonstrate variability in response, this time based on health characteristics coming into the trial. The next example comes from the VITAL trial, which was a 2x2 factorial trial testing daily supplemental doses of 2,000 IUs of vitamin D3, marine fatty acids, both or neither, on several health outcomes. And here we see that for one of the primary outcomes, there was no intervention effect on invasive breast cancer of any kind. A recent subgroup analysis, however, examined total metastatic cancer and cancer mortality as the outcomes. And here there was a very strong protective effect for the study supplements, for the vitamin D supplements, for those who began the trial with a BMI of less than 25. Again, variability in response, this time based on participant BMI. And my final example for this principle comes from the CARROT trial that I mentioned at the beginning. This analysis was for prostate cancer, which was one of the secondary cancers examined in the trial. Participants were allowed to enter the trial using their own supplements, which were usually multivitamins, as long as the doses of beta-carotene and retinol were low and no more than usual doses available at the time in over-the-counter supplements. We found that overall, men who came into the trial using any other type of supplement, which again was usually multivitamins, plus the carrot supplement, were randomized to the carrot supplements, had a prostate cancer odds ratio of 1.26. We found no association with non-aggressive cancers, but for aggressive cancers, we found that men who were using both any type of supplement, again, usually multivitamins, plus the carrot study supplements compared to placebo, had a prostate cancer odds ratio for aggressive disease of 1.52. This suggests that the dietary supplement habits coming into the trial invoked variability in response and subsequent disease risk. So at this point, you may be wondering why I'm showing so many subgroup analyses to dietary intervention trials. Well, the answer is that it's very informative, and it may help us think deeper about who responds and who does not respond, and it should guide us in precision nutrition interventions. I don't think that we should dismiss these subgroup analyses. So what have we learned so far? We have learned that there's interindividual variability in response to dietary interventions. And this variability is dependent upon study participant characteristics related to body habitus, usual diet prior to the intervention, dietary supplement use, and health characteristics. And I gave the example of hypertension. So future study designs, including for precision nutrition, should consider these lessons learned. In spite of these findings, I certainly do not take subgroup analyses lightly, and we need to consider the following. We have to consider whether or not the subgroup findings were by chance. We must consider multiple comparisons and multiple tests. And I will reiterate again that the WHI findings that I presented had very conservative Bonferroni tests applied. So we need to consider that when evaluating subgroup analyses. Measurement error is very crucial to consider, and this is particularly important when assessing baseline diet from self-report. We have to consider the phenomena of regression to the mean, which is all too common in some dietary intervention studies. And finally, we have to consider that when one component of diet changes, other components also change, and this adds to the noise. An obvious example is when macronutrient distributions change. For example, when fat as a percent of energy is lowered, something else has to increase, one of the other macronutrients, fat or carbohydrate. How does this affect findings? And phytochemicals are a more subtle example. So where do we go from here in precision nutrition interventions? What do we measure? Diet is extraordinarily complex. It's nutrients, dietary patterns, dietary supplements, eating frequency and meal timing, other eating behaviors, and how do we measure it? Self-report or biomarkers. In thinking about moving forward in terms of precision nutrition and how to account for inter-individual variability, I'd like to borrow a concept from the socio-ecological framework, which has been a useful model for population nutrition and public health in general. It tells us that there are numerous determinants of nutritional status and intake at the level of the individual, family, community, and public policy. Nutritional intake is not just a matter of individual knowledge or choice. Using that type of thinking, I'm wondering if we can think about the nutritional exposome. Measuring just a few nutrients or a list of nutrients is probably not sufficient. And based on what I showed earlier in the presentation, we need to account for other determinants of inter-individual variability in response. These characteristics range from usual dietary intake pattern or dietary supplement use habits to blood biomarkers, including integrated measures such as metabolomics and health characteristics. And I will close by mentioning a few gaps and opportunities. Variability in response to dietary interventions is quite prevalent. Supporting evidence comes from thoughtful, stratified analyses and well-conducted trials. One gap that's worthy of mentioning is understanding apparent non-responders could be informed by appropriate subgroup analyses, while importantly avoiding data dredging. And an opportunity is that we could develop an integrated measure of the nutritional exposome that will inform studies of precision nutrition. Thank you.
Good afternoon. It's a great pleasure to speak with you at this NIH workshop in relation to precision nutrition in relation to immune system status and inflammatory responses to diet. My understanding of the current state of the art in relation to diet and nutrition is that we've got very strong prospective data with nice and very elegant mechanistic insights in relation to immunity and inflammation. However, the data in relation to human dietary interventions is relatively weak, which may be ascribed to a number of variables, be it genetics, physiology, metabolic phenotype. But within the context of precision nutrition, it would be very nice to be able to progress our understanding in relation to what determines response or non-response to effective dietary interventions in the future. And I think that was one of the biggest gaps that we need to fill over the next 5 to 10 years. I think over the last year, I don't need to convince anyone that immune system status and our ability to mount an effective inflammatory response is a very significant public health issue. It's important to acknowledge that the immune system status is determined by a number of factors and variables out of our control, one of which is nutrient intake, which of course is within our control, as well as age, obesity, the gut microbiome. I can't touch on any of them, but something like the gut microbiome is very important in terms of, again, configuring the immune system and the nature of the inflammatory response, and also being a good marker of dietary and therefore microbiome diversity. The work my team has focused on has been in the area of chronic inflammation. Again, it's a very classical hallmark of obesity, type 2 diabetes, and some obesity-related cancers, which are very important and an emerging area for research. And it has been demonstrated that that chronic inflammatory state really does interrupt adipose biology, skeletal muscle biology, which is important in relation to insulin resistance and type 2 diabetes, but also interacts with immune cells and atherosclerosis, again, precipitating cardiovascular disease. Indeed, when I started in this area, there was a very simple one-way relationship between inflammation and metabolism, wherein, for example, in the state of obesity, where there's expansion of adipocytes, the infiltration of pro-inflammatory immune cells in adipose tissue, wherein those cytokines, be it IL-1, TNF, interrupt key metabolic pathways such as insulin signaling, and therefore lead to disrupted metabolism. However, data from the immunology field, which, for example, in this study, which integrated both metabolomics and transcriptomics, have shown very nicely that there is a very intricate two-way relationship between metabolism and inflammation. And indeed, this data from the immunology field shows that the metabolic status of the immune cell really determines whether it's pro-inflammatory or anti-inflammatory. And it really will be very important to understand this within the context of diet and nutrition, because while reconfiguring macrophage metabolism or T-cell metabolism may be a therapeutic target, it is very important to appreciate that diet is a key source of endogenous metabolites. So it will be very interesting to understand if and how diet can reconfigure that state of oxidative phosphorylation, which is associated with the resolution of inflammation, or pro-inflammatory responses, which are associated with the glycolytic state and fatty acid oxidation over the next coming years. My team is focused very much on the interaction between, say, different fats and one particular cytokine activation, IL-1, which is regulated by the NLRP3 inflammasome. For example, this work that we did comparing the impact of monounsaturated versus saturated fats. In this work, we were able to demonstrate that saturated fats are pro-inflammatory. They activate IL-1 through a metabolic sensor, AMP kinase, and therefore regulate insulin signaling and also adipogenesis. But the most important, or the reason why I'm showing you this slide, is that whilst you might get quite significant differences in, say, the amount of IL-1 secreted from adipose tissue or the amount of IL-1 released by the immune fraction in response to ATP, when we looked at this differential between the diets, looking just at fasting plasma levels, we do not see this big difference. You only see it when you get down to the individual organs or tissues. And so it's very difficult to sometimes just look at that inflammatory response when we're reliant on simple plasma biomarkers. But you can use very simple tricks. For example, manipulating either the fed or the fasted state, which these investigators did, wherein they quite clearly demonstrate that the level of IL-1 released in the fasted versus the fed state is very different, and you get enhanced pro-inflammatory responses, of course, in the fed state. And this can be very useful in terms of looking at difference between type 2 diabetics and healthy controls. And again, you need metabolic stresses such as ATP and also fatty acid levels to be able to show or to induce a stress and to show the difference in the inflammatory response between the two different cohorts. But of course, IL-1 is only one cytokine of many, and that's why we have used transcriptomic responses or signatures to give us greater understandings of the interrelationship between habitual diet and also, say in this case, adipose tissue biology in subjects with the metabolic syndrome. In this study, we compared the transcriptomic signature of the adipose tissue and integrated with dietary data and also dietary biomarker data, for example, plasma N3 PUFA levels, and also biomarkers of health, such as prostaglandin levels, triglyceride levels. And we were able to map clear networks wherein PUFA and MUFA intake were related to key inflammatory signatures and oxidative stress signals. And there was crosstalk between, say, triglyceride metabolism, lipid signaling, and also adipogenesis, which was regulated particularly by inflammatory markers. Along that vein of defining new biomarkers of metabolic health and inflammation, PBMC transcriptome can also be very useful. And we use this to quite an extensive extent within the context of a M.P.H study, which was called the Metabolic Challenge Study, wherein we completed metabolic challenges using either oral, glucose, or lipid tolerance tests in over 200 individuals. And we wanted to look at the repeatability between the lipid tests, the glucose tests, and then also compare either lipid versus glucose challenges. And that gave us a lot of interesting insights. As you can see here, this represents the oral glucose or the insulin response test in either young lean individuals in green, older lean individuals in yellow, or old overweight individuals. And you can see you get a much greater response, of course, in the postprandial state. And when we looked at the transcriptome signature of those individuals, particularly we found that the oral lipid tolerance test was particularly good at eliciting that pro-inflammatory response, where you got elicitation of the pro-inflammatory cytokines, and you saw the attenuation of the anti-inflammatory or the resolving arm with age. And these papers clearly show that we were able to use the transcriptomic response in terms of understanding the impact of BMI, the impact of gender, and that interrelationship between metabolism and inflammation. In relation to dietary interventions and precision nutrition, it really is very important to be able to look at the impact of the right diet for the right person at the right time. And very much this variability is quite controversial. Some people propose that that variability is the answer in relation to why some dietary interventions work or do not work. Others will propose that sometimes it's an excuse that essentially the dietary intervention does not work. But it is very important for us to understand if and how habitual dietary or metabolic versus inflammatory determinants determine response to non-response to dietary interventions. I would propose to you that it is relatively easy to demonstrate the bad effects of food, of say saturated fat challenges. What is more difficult is actually to look at the resolution of inflammation. Indeed, the public is very interested in foods that fight inflammation, but it's not very easy to do these types of studies. And indeed, maybe we have to rethink the way in which we do these types of studies. I wouldn't like to criticize somebody else's work, so I'll criticize my own, as it were. So this was an intervention study we completed in obese teenagers. It was an RCT using an anti-inflammatory nutritional cocktail, so a blend of N3-pufaz, alpha-tocopherol, green tea extract, and also lycopene, with a view to understanding whether or not this anti-inflammatory intervention could improve insulin resistance. We used a cocktail because, of course, there are a number of pathways that are interconnected in the inflammatory system, and it's very important to manipulate as many of those as possible. It was an RCT, double-blind, placebo-controlled, but we double recruited in order to be able to look at responders versus non-responders. Our cohort were overweight, very clearly, and also we did get this highly variable response with respect to insulin resistance. 38% of our cohort responded to the intervention, and that response was not down to differences in compliance, because we measured biomarkers of compliance, and this N3-pufar levels clearly showed that the levels of N3-pufar were similar for responders and non-responders. But in terms of defining those determinants of response or non-response, clearly you've got the metabolic determinants, the background insulin resistance, adiponectin. Indeed, adiponectin showed to be one of the most important determinants of response and non-response, which is very important. But in terms of understanding the how and the why, we completed very extensive PBMC, molecular work, epigenetic signatures, metabolomics, and also some lipidomics. But what I would say to you is that with 2020 hindsight, I would have completed this study in conjunction with a metabolic challenge test, because sometimes looking at just that fasting element is insufficient. In terms of key areas of innovation, I really would like to understand whether the resolution of inflammation is possible and to what extent. And the reason why I say this, and I'm taking data from animal studies here, but essentially this is a nice study which demonstrated that the innate immune system, which in theory is not trained, but it does have memory. In this study, they fed mice, and I'm sorry, we don't have human studies in this space, but they fed them a Western type diet, and you saw that you got a pro-inflammatory response systemically, but also in the bone marrow. But when they reverted those mice to, say, a healthy diet, even though the systemic inflammatory response went back down again, however, the bone marrow were still primed and we're still in this pro-inflammatory response. So within the context of human nutrition, I think it's important to address this. Also, Melinda Beck and her team demonstrated that unfortunately, again, this is a mouse study, but it demonstrated that while obesity induces T-cell metabolism reconfigurations such that the T-cell response is impaired, unfortunately, it was not possible to revert that with weight loss. Again, we need more clarity in relation to this in man. In relation to another key area of innovation, it is in terms of understanding how we can establish innate immune functionality. There have been a lot of work in relation to, or controversy in relation to vitamin D status, zinc status, also selenium status in relation to immunity in relation to COVID-19. And it's very important to, one, understand the interactions between those micronutrients, but also understand the interactions with other components of nutritional status, as in, how is it different in an obese individual versus somebody who has got protein energy malnutrition? The biomarkers in this space really need to be improved. We need better tools to capture cellular specificity because of course, we have so many different cells which function in different ways, and we need that granularity. But at the same time, we also need tools to look at continuous inflammatory monitoring. This is a nice tool which detects cytokine levels in sweat. And this study was done within the context of inflammatory bowel disease, but perhaps within the context of either innate immune training or chronic pro-inflammatory responses to diet, we could use novel technologies such as this. And so I hope that I've given you an idea in relation to some innovations that we do need in order to understand optimal immune and nutritional status and to have more effective precision nutrition approaches. And with that, perhaps we will have more or stronger human nutrition intervention data, such that we can improve risk, prevention, and also efficacy of diet interventions that we could be better able to improve and really accelerate the whole area of precision nutrition. Thank you very much.
Good afternoon. I am Frank Scheer from Harvard Medical School and Brigham and Women's Hospital. It gives me great pleasure to talk to you today about the importance of sleep and chronobiology for dietary responses. And I would like to thank the organizers for inviting me to be part of this exciting workshop. My talk will be composed of three parts. First, I will talk briefly about sleep. Second, the circadian system, and then melatonin and genetics. So for sleep, we've known for quite some time that disrupted sleep or short sleep are associated with increased cardiometabolic disease risk, including diabetes, obesity, and cardiovascular disease. And this is based on epidemiological data, but even prospective studies cannot fully provide the required evidence for causality. But these studies, together with in-laboratory experimental studies, have provided the evidence that these effects are most likely causal. And this is because the effect of short or disrupted sleep result in physiological changes that are in the pathway for disease development, including the increase of or the decrease of insulin sensitivity, insufficient beta cell compensation, and decreased glucose tolerance as risk factors for the development of diabetes. And while short sleep results in a slight increase in energy expenditure and would be expected to decrease body weight, this is overcompensated by increased hunger and appetite, and if people are permitted, people will overeat, which then results in a positive energy balance with increasing risk for the development of obesity. And there's even some evidence for experimental short sleep reducing physical activity, which of course also would contribute to increased energy balance. For cardiovascular disease, short sleep results in immune activation as well as increased blood pressure and sympathetic balance. But obesity by itself is also a risk factor for the development of obstructive sleep apnea, which perpetuates and further worsens the disruption of sleep. So closing this loop. What about the circadian system? Well, first of all, talk about the circadian system itself, then about its role in glucose control and in energy balance regulation. The circadian system is composed of a multi-clock system with the suprachiasmatic nucleus at the top, the master oscillator, regulating peripheral clocks throughout the body in virtually every tissue, organ, and cell. And this is enabled by a cell autonomous mechanism, a translation feedback loop consisting in all of the cells in the body that permit these cells to generate 24-hour rhythms. So if one places a fat cell or a skin cell in a petri dish, it will happily tick along with a rhythm of about 24 hours. And this clock system needs to be synchronized to the environment, and this is where light is the most powerful synchronizing piece of information for the circadian system, signaling through the retina to the suprachiasmatic nucleus the information of light, such that the system can be entrained to the light-dark cycle and also re-entrain, for example, after jet lag. But the circadian system then, the SCN then communicates the circadian information to the rest of the body using neural, humoral, and also temperature as time cues. But the SCN also regulates behaviors such as feeding, fasting, as well as physical activity, and these themselves act as zeitgebers or time cues to the peripheral oscillators, especially in metabolic tissues such as the liver and the pancreas. So what is the relevance of this to the topic of today? Well, the circadian system is important in the regulation of glucose. And to illustrate this, this is a classic work by F. and Kauter in Chicago, showing that identical test meals for breakfast, lunch, and dinner result in dramatically different glucose excursions. As you can see here, compared to breakfast, if we look at the dinner, there is an impairment of glucose tolerance, meaning the glucose levels rise much higher and glucose stays elevated much longer. Now, this has been known for quite some time and has been replicated. Our question was, to what degree is this regulated by the behavioral cycle and the environmental cycle, so external and behavioral influences on glucose control versus the effect of the internal body clock, the circadian system? And of course, there are many reasons to think that behavior and environment may be able to impact and cause this difference in glucose tolerance with an impairment in the evening compared to the morning. And this includes that breakfast typically is preceded by sleep, overnight fast, physical rest, and darkness, whereas dinner is preceded by extended wakefulness, shorter fasting durations, increased physical activity for longer durations, and light exposure. And so it could be that these behavioral and environmental differences are what is driving this. And so we investigated this, and this was work by Christopher Morris, a very talented postdoc back in the days. And this is what Chris did, is he ran a randomized crossover trial where people lived in their private time-controlled environment for 2 times 8 days straight in the CCI, and mathematically separated the influence of the behavioral cycle, comparing breakfast, so the first meal after the sleep episode, versus dinner, the last meal in the sleep episode, in the wake episode. And what Chris found was that glucose tolerance was impaired during dinner as compared to breakfast. And this was also associated with a slight decrease in pancreatic beta cell function, meaning that the insulin levels rise more slowly with dinner compared to breakfast. So there is an effect of the behavioral cycle. But to our shock and surprise, the effect of the internal body clock was much stronger, meaning that the difference between breakfast and dinner was much smaller, as compared to when we compare the internal body clock time with high glucose tolerance in the morning in green, where levels are quickly declining again, and impaired glucose tolerance in the biological evening, meaning that the circadian system plays a more important role than the combination of all the behavioral and environmental factors in our study. And likewise, also for early phase insulin increase, we see that the effect, this measure of pancreatic beta cell function, is more importantly influenced by the internal circadian system as compared to behavior. So the bottom line is that the circadian system plays a prominent role in regulating glucose tolerance, in part through beta cell function. What about weight regulation? We have provided evidence that late meal timing results in impaired weight loss success, both in dietary and in surgical interventions. So there was a 20-week-long weight loss intervention in over 400 participants, obese and overweight men and women, where the late meal, so the early meal, people who ate their main meal early lost about 29% more than the late eaters. And then subsequently, in a 6-year follow-up after bariatric surgery, we found that the late eaters were also showing impaired weight loss success. And this is illustrated here. So about 29% more weight loss in the early eaters, more successful and less successful in the late eaters. That wasn't explained by differences in dietary intake or self-reported physical activity or sleep duration, no differences in fasting leptin or ghrelin. Then in the same year, there was, of our first paper, there was a very nice publication where people, this was from Kubowicz and colleagues, scheduled either a large dinner and a small breakfast, or vice versa, a small dinner and a large breakfast, finding, again, consistent with the same concept of early meal timing being more advantageous for weight loss, that the late dinner group lost less body mass as compared to the, the large dinner group lost less body mass compared to the large breakfast group. And then in order to address the question of the importance of circadian timing, Andrew McHale completed a study in over 100 Harvard College students where they were tracking both the timing of their food intake with taking a photograph of each nutritional intake, as well as looking at their dim light melatonin onset, DLMO, which is a circadian phase marker. So the rise of melatonin in the evening is a reliable marker of each individual circadian phase. And what Andrew found was that the early eaters, meaning they ate their 50% of their caloric intake many hours before the start of the biological night, had on average lower body fat compared to the late eaters. And the same was true for BMI. And this was really further emphasized the importance of circadian timing, because when he instead used clock time, so instead of the circadian marker, you just use clock time, this association fell apart. So the conclusion is that circadian phase of food intake correlates with body mass index, and even experimental manipulation results in changes in body mass loss. Now, why might this be? Chris, again, this was a study that published 5 years ago, or 6 years now, found that diet-induced thermogenesis, so the approximately 10% of the daily calories you burn following the intake of a meal is under strong circadian control, where if food intake, the same identical intake occurs at the late endogenous circadian phase, results in only about half of the diet-induced thermogenesis as compared to the same meal eaten in the early circadian phase. So theoretically, eating late in the circadian phase could contribute to a higher body mass index. So this may be one of the mechanisms that may play a role, the circadian control of diet-induced thermogenesis. Of interest, we also found that hunger and appetite themselves are under circadian control with higher hunger and appetite at a late circadian phase in the biological evening. And Jingyi Xiang recently showed that this associated with higher levels of the hunger hormone acylated ghrelin, so the active form of ghrelin. Now, the last topic about melatonin and genetics. About a little over 10 years ago now, the melatonin 1B receptor gene, MTNR1B, had been shown to be a type 2 diabetes risk gene, so a particular variant associated with increased type 2 diabetes risk. Then, in collaboration with Mattei, we showed that melatonin, when taken orally 5 milligrams 15 minutes before an oral glucose tolerance test, reliably impairs glucose tolerance both when assessed in the morning and in the evening. We then showed that this was dependent on the participants' genotype for MTNR1B, where the impairment of glucose tolerance was only observed in the risk carriers. And subsequently, and this is illustrated here, together with Marian, Richa, Saxena, we found that physiologic concentrations of melatonin are also important for glucose control, such that if the dinner is delayed by 3 hours, that glucose tolerance is impaired, but it's only impaired in the GG carriers. And so when dinner is delayed, when such that melatonin levels are elevated, that it interacts with the genotype, showing a melatonin, a time of day times genotype interaction. So together, this really shows that melatonin signaling through the MTNR1B risk receptor and modulated by the genotype for this receptor is influencing glucose control. And so in summary, 3 hours that glucose tolerance is impaired, but it's only impaired in the GG carriers. And so when dinner is delayed, when such that melatonin levels are elevated, that it interacts with the genotype, showing a melatonin, a time of day times genotype interaction. So together, this really shows that melatonin signaling through the MTNR1B risk receptor and modulated by the genotype for this receptor is influencing glucose control. And so in summary, disrupted sleep has adverse metabolic effects. The circadian system, which is a multi-oscillator system, is generating intrinsic endogenous 24-hour rhythms, so even in the absence of environmental and behavioral cycles. And this impaired genes, so particular variants as the dinner is delayed by 3 hours, that glucose tolerance is impaired, but it's only impaired in the GG carriers. And so when dinner is delayed, when such that melatonin levels are elevated, that it interacts with the genotype, showing a melatonin, a time of day times genotype interaction. So together, this really shows that melatonin signaling through the mTNR1B risk receptor and modulated by the genotype for this receptor is influencing glucose control. And so in summary, disrupted sleep has adverse metabolic effects. The circadian system, which is a multi-oscillator system, is generating intrinsic endogenous 24-hour rhythms, so even in the absence of environmental and behavioral cycles. And this system robustly influences metabolism. I've shown you examples for impaired glucose tolerance and pancreatic beta cell function, as well as lower diet-induced thermogenesis with relevance to body mass regulation. And finally, melatonin, both pharmacological and physiological concentrations impair glucose tolerance, especially in mTNR1B risk variant carriers. And so a lot of this latter work really shows, and of relevance here, is that it's not just important what you eat, but also when you eat. And so in this regard, just a few knowledge gaps that I wanted to address is the importance of developing standardized study procedures for the control and/or assessment of sleep, circadian phase, meal timing, melatonin, and genetics in nutrition research. So to take these factors reliably into account. One of the challenges is the assessment of meal intake. I showed you participants taking pictures, but it would be good to develop passive assessments. Continuous glucose monitoring is one example. Visual monitoring, which is more challenging, but even new developments for assessment of swallowing, etc., would be of value. And then the importance of determining the relative contribution, but also the underlying mechanisms of the effects of sleep and circadian biology in nutrition research, I think remains a key focus point. And then I think there is great promise in the development of interventions based on this new knowledge, so evidence-based, for example, meal timing interventions to improve metabolic health, as well as interacting with genotype to allow personalized interventions in nutrition research. And so with that, I would like to thank all my collaborators, the members of the MCP, the funding agencies, and you for your attention. Thank you.
Good afternoon. I am Josiemer Mattei, Associate Professor at Harvard Chan School of Public Health. And in this section, I will discuss what are the social determinants of health, how are they a main source of disparate resources to health, why they matter in precision nutrition, and how we can overcome main challenges in this field in order to improve health for all individuals equally. Social determinants of health, which you will see referred to as SDOH throughout the presentation, are the conditions under which people are born, grow, live, work, and age, and include factors such as socioeconomic status, education, employment, social networks, and neighborhood characteristics. We often think of social determinants of health as the economic and political structures, the social and physical environment, and the access to health services that shape a person's health and well-being. They are often referred to as upstream factors because they mark the start of an individual's characteristics that lead to the downstream health outcomes. Social factors are important because they explain most of an individual's health. While specific estimates vary somewhat, in general, the socioeconomic and environmental factors shape around 40% of a person's and population's health, having a larger impact than other factors like biology and behaviors. And recognizing the social factors that influence health is a responsibility for all of us in healthcare, public health, and social justice. Only by deeply accounting for these factors can we fully and better address resource allocation and population health. The other important aspect of social determinants is that they expose and explain health and nutrition disparities. Social factors, especially poverty, structural racism, discrimination, housing conditions, are primary drivers of health disparities. We can think about this using Links and Phelan's fundamental cost theory. Individuals with wide range of flexible resources with regards to knowledge, wealth, power, and social networks can afford to focus on their health. But those lacking these resources need to spend their effort in acquiring them to begin with before even being able to care for their health. So fundamentally, who dominates the upstream factors that determine dietary choices is unequally distributed within the population. And the people unfavorably impacted are groups vulnerable to the inequitable resources. So let's illustrate how income inequities impact nutrition. The authors of this study use a national representative samples of adults from M.P.H to investigate trends in dietary quality from 1999 to 2010 within socioeconomic groups defined by formal education attainment and poverty income ratio. They first found that for everyone, diet quality increased throughout the study period. However, when the trends were stratified by socioeconomic status, the improvement only remained for those in the high socioeconomic status. Not only the diet quality was lower for people of low socioeconomic status, but this discrepancy widened over time. So the inequality gap increased over the course of a decade. As an aside, I used its title in reference to the media headlines that the study generated. But I want to point out that we have to be mindful of the way that social determinants are labeled, as they may entrench patterns of exclusion and power. Labels such as poor, uneducated, low class, disabled, old, instill a sense of inferiority and should not be used. So thinking deeper about what challenges do people with low income may face when choosing their diet. And one potential source is where they live and access food. And at the forefront of these are food deserts and food swamps. Food deserts are areas that lack sufficient and affordable healthy foods, while food swamps describe areas teeming with nutrient-poor, energy-dense, unhealthy food options. People who live in these areas generally have lower diet quality. But the bigger issue is that people of low income, as well as racial ethnic minorities, are more likely to live in deserts and swamps. And this has deeper roots in historical structural racism and discrimination, discriminatory zoning laws, and even biased education systems that restrict socioeconomic growth. Despite knowing this, it is very difficult to intervene or regulate on the deep causes driving food deserts and swamps. So public health nutrition approaches are a more band-aid approach, patching the system with strategies such as incentives for supermarkets or delivering food produce, but neglecting the deeper socioeconomic and structural embedded disparities. And thus far, the impact of interventions on food deserts in reducing disparities is weak. So how this all fits in precision nutrition? Well, precision nutrition is the concept that one size does not fit all. It leverages human individuality to drive optimal nutrition recommendations that prevent, manage, and treat disease. In that spirit, precision nutrition should use all relevant information on an individual's characteristics, including social determinants, to refine disease risk stratification. But that is the ideal situation and not the norm. And in fact, it has been questioned if it's even feasible for an individual-centered precision nutrition approach to replace general population-based dietary counseling. Instead, a more feasible application of precision nutrition in practice would be to counsel and assist a patient following general dietary health, still accounting for individualized data, if available, but aligning, making sure that it aligns with the social and cultural background of the individual. Again, that's ideal. Precision nutrition approaches often fail to consider or even collect information on social determinants of health. Machine learning algorithms can integrate behavioral data, omics data, biological parameters, anthropometrics, spit out the best diet for a person's optimal health. They rarely include data on a person's social and environmental characteristics. And even when they do, translation to vulnerable communities is sorely lacking. One challenge is that there is no consensus as to which set of factors define social determinants of health. Several organizations have put forth definition, and there are some commonalities, such as placing socioeconomic determinants at the forefront. But there are some differences. Food-related factors are differently classified depending on the framework, sometimes as economic stability, sometimes as neighborhood and built environment, and sometimes as its own category. One agreement across factors is the complexity of interaction among social factors, although they don't necessarily agree on how to map these complex links. One safe factor can be considered a root cause in one framework or a mediator in another framework. And these discrepancies have contributed to a gap in understanding the mechanisms linking the social factors to disease burden. Moreover, that complexity of multifactorial pathways across social determinants makes their analysis difficult. Not only are the models complex, but they also need to be precise. We have multiple factors often measured in different ways. So how do we measure an income construct? Is it income as poverty? Yes, no. Income of the person, income of the household, subjective social status perceived by the person. So these challenges have limited establishing mechanisms and causality, which then limits their applicability in precision nutrition. So we need precise, harmonized, unbiased, complex system modeling to examine the complex social systems linked to health disparities. Another large limitation is that studies that may inform precision nutrition remain largely limited in representation. This recent systematic review evaluated the effect of precision nutrition of randomized clinical trials and found that while promising in improving diet quality, there were clear limitations in representation. Most studies were comprised of females. All of them were conducted in US, Canada, or Europe. There were no studies on younger or vulnerable populations, and most studies had short duration, raising the question of sustaining tailored dietary advice throughout the lifespan. So precision nutrition studies need to have representation from wider racial, ethnic, and vulnerable populations that face the social inequities. And precision nutrition interventions should consider these populations' social dynamics, cultural preference, and socioeconomic conditions in order to be successful. Another challenge is how to incorporate social determinants in precision nutrition is that individuals process these factors differently. Embodiment refers to how we biologically deal with the material and social world around us, how these social inequities get under our skin. A person may be exposed to adverse events in their lives, chronic stress, and their body's physiological system may respond by raising hypervigilance, blood pressure, inflammation. And this wear and tear that accumulates in the body over time is referred to as allostatic load. However, each person's physiologically integrate these responses in a different way. This is the same method, the same way that epigenetic modifications and metabolome and microbiome responses operate. However, we know very little as to how social determinants ingrain in the body. And two factors that limit this is how to precisely measure the timing from social exposure to physiological response, and how to differentiate what may be normal processes versus disparities-driven processes. One last gap in the research is that it has focused mostly on adverse social factors. But despite having the same exposures to adverse social determinants, not all individuals or populations experience worsened health outcomes. Protective social factors such as optimism, social support, sense of purpose, positive coping, resilience may prevent or buffer the harmful effects. And two key research questions that remain are, to what extent do protective factors explain differential responses to social stressors? and which positive factors can we leverage to overcome social inequities and optimize precision nutrition. So in summary, we have several gaps to close in order to fully embrace and understand the role of social determinants on precision nutrition. From neglecting the issue altogether, lack of consensus on how to measure and define it, the complexity requiring advanced analytical technologies, and the limited data in diverse population. We also don't fully understand the mechanisms between upstream and downstream factors, or how embodiment and protective factors operate. And one last question that remains is the potential for sustainability, clinical reach, and implementation of precision nutrition interventions that do account for social determinants. On the other hand, this opens many opportunities and next steps to close these gaps. Rather than focusing on band-aid approaches that patch the downstream system, we should work at treating the upstream issues. One key to fill the research gaps mentioned previously is having adequate funding that prioritizes social determinants in precision nutrition. Similarly, a focus on social determinants should be taken when designing research studies and promoting health policy. Nutrition research should carefully consider not only the dietary intake, but also the social and cultural norms that drive dietary choices, including food access and security. Policies that mind the upstream factors can include wider healthcare coverage, easy access to nutrition assistance programs, filling the educational gap at younger ages, anti-poverty programs, and anti-discrimination laws. This may be facilitated by better educating and training researchers, nutritionists, clinical practitioners, and public health stakeholders at all levels on the importance and merits of addressing social determinants. We have an opportunity to leverage and optimize the use of existing cutting-edge tools and databases to collect, share, and harmonize data. And we also must prioritize the inclusion of underrepresented and vulnerable groups that are prone to the social inequities. This all may require strong multi-sector partnerships. I should note that several organizations have put forth recommendations and resources on how to research and intervene on social determinants for optimal precision nutrition. And I include some of these references at the bottom of the slide. I want to end with a quote from Sir Michael Marmot, a vocal advocate for closing the gap in health disparities. Health inequities and the social determinants of health are not a footnote to the determinants of health. They are the main issue. As we move forward, we must remember that health is more than just an absence of illness. It is a resource that enables us to live a valued life, and everyone deserves to have these resources equitably. Thank you.
Hello, everyone. I'm Cheryl Anderson, a professor and dean of the Herbert Wertheim School of Public Health and Human Longevity Science at the University of California, San Diego. This presentation covers psychosocial and cultural factors that influence dietary intake and dietary patterns. And it's very closely related to the presentation you just heard by Dr. Mattei, which focused on nutrition-related health disparities, socioeconomic influences, and social determinants of health. This is an extremely important time to focus on psychosocial and cultural factors, as we are at a moment in history when we're facing a pandemic of SARS-CoV-2 and the resultant COVID-19. There are high levels of anxiety for many. There's a looming economic crisis, joblessness, feelings of hopelessness, and a really important national conversation that is happening about something that those of us who work in public health know all too well. Racism is a public health crisis, and the conversation that we're having about structural and systemic racism, particularly anti-Black racism, has implications for how we think about diet and nutrition. Before we delve into this talk about cultural factors, let's take a moment to define what is culture. It's broad, and so I like these two definitions. One's Nancy Krieger's that says it indicates a particular way of life, whether of a people, a period, a group, or humanity in general. Cultural variables such as spoken language, shared norms, beliefs, and expectations, and behavioral customs can be potential targets for healthcare intervention, as Fisher outlines. It turns out that as we think about cultural influences on dietary intake, we're talking about what's normative. What are the behavioral customs around foods that should be or could be potential targets for nutrition interventions? For example, is it cheap and convenient to eat healthful foods? Is that our culture? I love this quote, so I'm going to throw it into the talk at this point, just as a last bit of inspiration. Farming looks mighty easy when your plow is a pencil and you're 1,000 miles from the cornfield. I often remember this as I think about psychosocial issues and cultural issues, because they don't often fall into that mechanistic framework that we think about when we're thinking how diet might influence health outcomes. And I'm going to argue that they absolutely should. So here's a framework that we might use to describe the mechanisms by which we think diet impacts health outcomes. And I'll use cardiovascular disease here as an example. So we could argue that diet has major implications for our lipid levels, blood pressure, endothelial function, insulin sensitivity, and these and other risk factors impact whether or not we end up with cardiovascular disease. Well, in thinking about psychosocial and cultural factors, we're taking one step upstream to really better understand what are some of the things that could impact us having the chances of having a healthful diet. So for the purposes of this talk, these are factors that include stress, social support, hostility, depression, hopelessness, unspoken language, the norms, beliefs, especially misinformation that is a real concern right now in our society, expectations and behavioral customs. These things have a bidirectional relationship with whether or not we achieve a healthful dietary pattern. And that pattern, as you can see outlined here, would include a variety of vegetables, fruits, whole grains, low-fat dairy products, low-fat protein products, healthful oils, and being limited in saturated fats, added sugars, and sodium. So when we think about the mechanisms by which diet impact outcomes, like cardiovascular disease, for example, it's really important to look at the upstream factors that could also be at play and to include those in not only how we think about proposing the work that we're going to do, but in the execution of the work that we're going to do. So why is this important? Well, we know that there's increasing population diversity, and this map shows prevalence data from a special report from the 2000 census. We can see that in some counties, the group with the highest percentage of the county's population is Native American. We see that in cream. Latinx, we see that in green, and African American or Black, we see in purple. And this has been happening now for decades. We had a past former director of the US Census Bureau who was quoted as saying, "We're on our way to becoming the first country in history that is literally made up of every part of the world." And diversity is more than just identifying where we have growing prevalence of racial and ethnic groups, but it's really about the appreciation for the different cultures that we bring. Now, an important concept when thinking about culture is acculturation. And this figures in a acculturation model that tells us that when a group or a person is acculturated, then behaviors and beliefs reflect the home country. As a person or a group moves toward becoming acculturated, then behaviors and beliefs reflect the host country. And when there's true diversity and acceptance of culture, then there's less demand for acculturation, for us to move from where the acceptance and belief systems have been in the home country to the current country. And I've heard this decreasing demand really nicely described as we're no longer looking to be in a melting pot. We have more of an interest in being in a salad bowl. So we know that risk factors as well as intervention success have been shown to differ by race ethnic groups. And as shown here in these data from the Trials of Hypertension Prevention and the Hypertension Prevention Trial, weight change by months of follow-up in the study varied by race. These are data for Black females and white females. And we see data from the Hypertension Prevention Trial for Black women, for white women, from the Trials of Hypertension Prevention for Black women, and then for white women. And similarly, when we look at males, we also see similar differences in success with weight loss, even though it's not as stark as it is for females. And the authors, Shriki Kumanika and colleagues, suggested that racial differences in weight loss may result from a combination of behavioral, sociocultural, biological, and programmatic factors. I want to touch base briefly on some of these sociocultural and behavioral factors that may have been at play here. So when we're thinking about the promotion and adoption of healthful dietary patterns, we can think about this at the individual level, at the provider level, and at the level of the healthcare system. And as these factors are operating in these various spheres and we're conducting nutrition intervention studies, we should be thinking about how they're not only going to interfere with our interventions, but also with our outcomes and the sustainability of the impact of any changes that we accomplish in the research setting. So for example, on the individual level, we could have traditions, distrust in medical information, the availability and access of foods, lack of social support, life stress, literacy, numeracy being at play. At the provider level, not having enough time, not believing that the advice that is given would be adopted by patients, or even lacking the cultural competence to engage in this encounter with patients. And at the healthcare system level, third-party payers, how we schedule, limited hours in operating, and staffing concerns could also serve as barriers. Now, these barriers could also be looked at and manipulated so that they become facilitators of healthful dietary patterns. So if we think about traditions and beliefs, there are those that facilitate good health, and they should be acknowledged and leveraged in order to assist in accomplishing better outcomes. The ability to use personal and neighborhood resources, problem solve, and establish trusting relationships with the healthcare professionals are included here as well. Now, a public health framework, the socioecologic model, can be very helpful in thinking about psychosocial and cultural influences. So this model has been adapted to further investigate how food and beverage intake and physical activity might be impacted by individual, behavioral, other sectors of influence, social norms, and values. And we see here that psychosocial factors get called out, as do some issues that we know that are likely quite cultural. And the bottom line, or the takeaway message, is that we can't have conclusions that are drawn about dietary intake that simply rely on individuals to be able to make those behavioral choices. Now, it's one thing to acknowledge that the factors are at play, but what are some of the strategies that could be used to address them? Well, I'm going to just point out a few things from 3 very large NIH-funded trials that were pretty successful in changing dietary behaviors. The premier study used strategies that included staff training to acknowledge cultural factors. They talked about the myth of race, did motivational interviewing, really understood socioeconomic and within group diversity. There were participant considerations, including ethnic foods, really thinking about culturally relevant activities and tailoring the approach across various study sites. In another large trial, the Diabetes Prevention Project, they matched the ethnicity of case managers in some cases. They gave curriculum in more than just English. They also used Spanish. They included ethnic foods and cooking methods, and thought about diverse options for how to facilitate maintenance of change. In the trials of non-pharmacologic agents in the elderly, some of the considerations included having heterogeneous learner characteristics accounted for, really thinking about vision, cultural diversity across the centers also. Something that has come to a scientific discussion recently is structural racism as a fundamental driver of health disparities. And I just want to briefly touch on this publication that happened recently in November. This figure really, I think, advances our thinking about psychosocial factors and cultural factors in showing the influence as far upstream as slavery, structures that have been put in place to maintain white supremacy, how that impacts CVD and stroke risk factors, toxic stress levels, and eventually may lead to poor cardiovascular and stroke outcomes. We also know that upstream factors are important for health. We see here that clinical care factors, health behaviors, physical environments, and social and economic factors account for varying amounts of influence on health outcomes. We would even argue that some of these things are fundamental structural systemic issues that drive differences that we see in outcomes. With regards to social and economic factors, they convert into simple things like food insecurity that we know is likely to definitely get in the way of accomplishing a healthful dietary pattern. We also have a culture of misinformation. The WHO has recently been trying to target this through a very coordinated and strategic effort. A webinar was recently held to try and address misinformation and data transparency, particularly as it has been happening with SARS-CoV-2, and we know that it happens in nutrition as well. So just to wrap up, the reason why we're talking about these factors, and I'm really looking forward to the panel discussion, is our eventual goal is social justice in health. We're not just looking for equality, meaning we're giving everyone the same support, or equity, where we're giving them the support that they need. But what we want to do in our research to have true impact is to think about justice. How do we address root causes and get them out of the way so that those systemic barriers, whether they be psychosocial or cultural, do not lead to adverse dietary intake. Instead, promote all of us, all citizens, being able to have healthful diets and healthful dietary patterns. Thank you very much.
Hi, this is Marian Neuhouser. I think we're having some technical difficulties. We're waiting for our moderator, Dr. Parks, to reconnect so we can have the Q&A.
Well, maybe while Elizabeth is joining us, we can start with the question and answer. Let me see. How far are we able to offer precision nutrition counseling in clinical settings?
Yes, hello.
Okay, are we ready to go? I'm getting a lot of feedback. But I'm okay. Are we ready to go? I'm ready to ask some questions if you can hear me. I'm getting a lot of feedback. Yes. Okay. I'm going to start with some questions for Dr. Mattei and Dr. Anderson. And the first question is that the models show that we have to collect a lot of information to make precision nutrition kind of treatments. And the question is, won't this large burden added to healthcare differentially impact different groups of people? Dr. Mattei? Yeah.
Yes, I'm unmuted myself. Thank you. That's a good question. There are two questions here. The first one is about the collecting the data and the feasibility and the burden of collecting, and then how to use it. For the first one, I don't feel that we have the lack of techniques or tools to collect the data. We can collect multiple. I'm seeing that you cannot hear me. Are you able to hear me?
I can.
Okay, sorry. I was seeing that comments that you couldn't hear me. Sorry about that. We have the technology to collect that. If we are able to collect information on multiple metabolites, on multiple in diet, we can definitely collect the data for these and process it and analyze it. So it's not the lack of technology to get it, it's really how to do it from an individual. And like I mentioned before, what exactly to get so that we can use it. And that's something that we don't know very well right now, which determinants and which factors are more relevant, which one we need to focus in collecting. The second question is, if we have this data, how can we use it to reach the vulnerable populations? Am I interpreting the question right?
I think so. Yes.
Okay, so that's the second question, and I think that's the biggest limitation about precision nutrition is who's reaching, what populations is it reaching, and that's right now, if we don't have data that applies to the vulnerable populations, we cannot even reach them. So to me, both comes hand in hand. We have to get the data from participants and from individuals who represent all diets, all responses, all individualities, so that we can use that right now. So it goes hand in hand, and until we get that data, we cannot implement it at the clinical level.
Yeah. And Dr. Anderson, would you have a comment? And I'd also, what I took away from your talk is that many studies look at a single nutrient or a combination of nutrients, and they're focused on a particular narrow field. But what you are talking about is that the social determinants of health and the environment almost affects everything. It's a fundamentally different way of looking at precision nutrition.
Yes, very nicely interpreted from the talk. So thank you for that. You know, we started the whole precision moniker with a precision medicine approach. And then I heard, you know, that shifting a little bit to a precision public health approach. And now I'm hearing the term precision nutrition. And I think in all of these spaces, it has become very clear that we can't get anywhere near precision without collecting quite a bit of information. And so let's not sell ourselves short as we embark upon precision nutrition in terms of getting information. Now, we have had quite a bit of advancement in terms of using technology, whether it's wireless or mobile, to help us get biological information. And let's figure out how to use that to now get information that is psychosocial, that is cultural, that is behavioral, because these things, as I hopefully communicated well in the talk, they are those upstream factors that once we're finished in the ivory tower, everyone returns to be faced with those factors. And if we're going to have sustainable change as we embark upon this precision nutrition initiative, we must be mindful and we must find a way to make this happen.
Yeah. One environmental effect of sleep that brings us to this issue that sleep affects many things. And as I understand it, we've looked at modifying sleep and looking at metabolism. Dr. Scheer, how would you flip that on its head to think about the future? I'll need you to unmute, I think. Sorry.
Sorry, I have to apologize. Due to these technical difficulties, I was out for a bit. Could you repeat the question, please?
The question is that we were focusing on how the environment affects metabolism and, for instance, socioeconomic status or something. But up to this point, we've thought about altering sleep and then looking at its effect on metabolism. How would you kind of flip that idea on its head to go forward?
Right. Well, I think one question I often get from especially more general audiences is, well, what about the other way around? What is the effect that diet may have on sleep, as well as the circadian system? And despite the great interest in the general community, there is surprisingly little research that has been done, experimental, high-quality research. And so there are a few smaller trials looking at both timing and also content of dietary intake. For example, there's one study looking at high GI rice at the same time before scheduled bedtime, showing slightly faster times for falling asleep. But it's really a gap, I think, in the scientific community. The other interesting side of this is the relatively recent discovery of the effect of high-fat diet in rodent studies, where this leads to disruptions of circadian organizations, changes in timing, blunting of rhythms. And so this is actually something we're currently very interested in, but haven't studied yet. So watch this space.
Yeah, thank you. I'd like to follow that up with this question about some people, when they get stressed, gain weight. Some people lose weight when they get stressed. Does everyone in general increase their food intake to stay awake? Do you find that to be a...
Well, I think there are, I don't know direct evidence of high-quality studies that have looked at this specific question. However, it is true, and this has been shown by many studies, that if you truncate sleep or you disrupt sleep otherwise, that people will report increased hunger and appetite. There will be increases in the hunger hormone ghrelin, decreases in the satiety hormone leptin, and if people are permitted, they will eat more, which is what I kind of showed in my review. So that is maybe a link between short sleep and then people feeling more hungry. A second aspect is that we've shown that not just is hunger dependent on how long before you've eaten, but actually the circadian system has a role in regulating, modulating hunger and appetite. Surprisingly, actually showing a peak at around, circadian phase equivalent to 8 p.m. and a trough in the morning. So also that if you're awake for longer in the evening hours, you're exposing more of the circadian phase when hunger and appetite are elevated.
Yeah, thank you. Dr. Neuhouser, how would you define a responder or a non-responder? How do we think about that generally?
That's a really good question. I think if we set out with a hypothesis to test about a particular dietary intervention, and we have good preliminary data and good biological rationale to posit that most individuals should respond in, Y manner, that would suggest a responder.
Yeah.
I think what we've observed and what's been presented so far today is even if we have that very firm biological rationale, whether it's based on preclinical animal data, observational data, other data to support the scientific premise, there's still some people who don't respond. And I think in the past, it may have been thought, oh, these people just didn't follow the protocol, or they were non-compliant, or on and on. And I think what we're learning, which will be helpful in designing future interventions, is that it's not a one-size-fits-all. For example, in the Vital trial, do people with a higher BMI who didn't have a lower cancer mortality, do they perhaps need a higher dose? In other studies, what are the other factors? All these factors, we can't boil it down to just one or two factors that influence who is a responder, and that's why we need these comprehensive nutritional exposome, measures to help us further understand who's a responder.
Yeah, and this brings me to my question about measurement error. So how do we think about that with respect to figuring out if someone's a responder?
Yeah, so measurement error, particularly with dietary self-report, is a very well-known phenomena. It's mostly identified with energy and some macronutrients. The extent to which measurement error affects overall assessment of dietary patterns in self-report is less well known. Biomarkers, the biomarker signature of a diet is something that we're working on right now, and I think we've identified some biomarker signatures that reflect overall dietary patterns, especially for reflecting, say, the healthy eating index. A limitation of biomarkers is that it's very short term. Whereas when we ask for self-report, we can ask for a longer term diet. So again, this is why we need it all. We can't rely on just one type of measure to understand and design studies.
Yeah, thank you. Dr. Scheer, I'll go to our chat. Someone's asked a question. They said, we know that teenagers, especially older ones, often have upside down schedules in that they stay up very late and sleep very late if they can. At some point, this behavior right-sides itself. Should we conclude that these teens are at risk for cardiovascular problems in life, or is this a sociological phenomenon or a hormonal phenomenon?
So I presume that question is for me.
Yeah.
And I briefly showed one recent study of Andrew McHill where it is, and this is cross-sectional, so causality is hard to draw from that, but we do see that, two things. One, yes, it's very clear that in teenagers, the preferred time, with other words, chronotype, is becoming later, peaks at adolescence and then becomes earlier again. So that's one could consider normal response in the sense that also animals show the same trend. However, within the teenagers, if you do look at when they eat relative to their internal circadian clock, so even though that whole clock might be delayed, but if their mealtime is relatively more delayed than their internal central clock, then we see this associated with increased adiposity, increased BMI. And I think future studies, longitudinal studies, are needed to determine whether this has hard endpoints such as increased risk for the development of obesity and cardiovascular disease.
Yeah, Yeah, thank you. Dr. Anderson, there's a question to you. Some future studies aimed at determining factors influencing interindividual variability in health outcomes may use controlled feeding study designs where all foods and beverages are provided to subjects. Should we be measuring cultural psychosocial factors in these controlled feeding studies? Can you discuss how we might use these data to better understand what's happening when volunteers are not self-selecting their own diets, which we know people don't like to eat prepared diets?
Yeah, thank you for that. In fact, I think we do this a bit already. So arguably, if you think about one of the most successful feeding studies, especially in the cardiovascular health space, the Dietary Approaches to Stop Hypertension study, there was intention around how the diets were prepared in that context. And by intention, I mean to reflect what we think of as a typical American dietary approach. And in the same vein, as I say, you know, we wanted to have not something that only the very wealthy or the very educated or those who have access to specialty grocery stores could achieve. We also now, I think if we were doing a study like that, would stop and say, okay, when we say typical American, what does that mean? Right? It probably means something more than the way that we were thinking about it back then. And so in the feeding studies that I've had the great honor of working with, we are talking about who are we attracting into the study, and if we want to have great retention, if we want to have good adherence, if we want to be able to just suggest to individuals that we want to attract that you are welcome here because you see yourself and you see a part of your culture here in what we're providing for the feeding experience, then this has to be done, right? So I think we already do it intuitively. It's how we're doing it and who we're saying we would like to be included in that word typical diet. Or, you know, because no matter if we're addressing hypertension or diabetes or sleep, you name it, whatever the risk factor is, we want everyone to have a good chance at being able to not just follow that diet while they're with us in the ivory tower concept. We want that diet or those patterns to be sustainable beyond the university walls or the research setting. And if we're going to have this kind of long-term adoption, adherence, maintenance, we must take these things into account.
Yeah, I think that's a great way to conclude this session. We're going to take a 15-minute break, and when we come back, we'll start with some talks again. Thank you all.